Thank you very much. So my name is Nikolaus Hermann-Spahn from the Comprehensive Nuclear Test Ban Treaty Organization in Vienna, and talking today about metrology in support of nuclear test monitoring. So I will briefly explain what our treaty is. Some of you may know this too well. Then describe our international monitoring system, and of course, with a focus on radionuclide monitoring. There's two aspects to it, this gamma spectrometry for aerosols, and for noble gas monitoring, we're looking at xenon. So there's two different aspects, and I'm trying to explain a little bit the metrology needs that we have. The treaty bans all nuclear explosions anywhere on the planet. So, and this is a very important tool for nuclear non-proliferation. And it's widely accepted. 187 states have signed the treaty. It's not yet in force, but in the meantime, we have set up international monitoring system and other tools to verify that no one is testing nuclear weapons. Of course, we cannot prevent it, as in the case of North Korea, but we will detect it. The international monitoring system consists of 320 stations dotted around the globe, like this. It uses different technologies, seismic, infrasound, hydroacoustic, but we will focus on the radionuclide component of this network. There are 80 radionuclide stations, and half of those, 40, are to be equipped with noble gas monitoring capability. So currently, the green dots are stations that are installed, contributing data to verification monitoring. The network is supported by 16 radionuclide laboratories, and I will explain a little bit about this later. We, of course, detect a large number of different, or trying to detect a large number of different fission and activation products. However, we'll mention barium-140 or lanthanum-140 a few times on the next slides. That is because it's one of our key nuclides. The reason is it has a high fission yield and reasonably high gamma emission yield, so it's one of our those nuclides that will be present in a nuclear explosion scenario spectrum. It's also important because it's one of our clock pairs that we can use with 13 two-day lifetimes. So with a detection shortly after a nuclear explosion, it allows us to determine the zero time. In addition, it's also a decay product. Barium is a decay product of xenon-140. So in a scenario where the aerosols are confined in an underground test, but the noble gases are released, we will still see barium-140 as a decay product of xenon-140. So with this radionuclide part of the network, we'll be trying to pick up even a small nuclear explosion, 1 kiloton, anywhere on the planet. Within 14 days, with a reasonably sparse network, it's 80 stations, but we have to cover the whole globe. So if we're looking at a 1 kiloton nuclear explosion, if it's an atmospheric test, talking about trace levels, that's like a Fukushima type release, that's not challenging. The challenge is to detect underground tests where the releases are much reduced or even zero. But for certain scenarios, we come up with a requirement on the network in terms of detection limits. Of course, they are usually volumetric detections, but so the way we're looking at microbecquerels per cubic meter or millibecquerels per cubic meter, but for the purpose of looking at the requirements for the sample sizes that we have. So for barium, it's less than a becquerel that we need to detect. And for noble gases, xenon-133, it's like 10 millibecquerel. That's kind of the magnitude we're looking at. So when it comes to aerosol monitoring, that's nothing special. We just have standard gamma spectrometers at the stations. And there the challenges are more around sample inhomogeneities, variations in the sample geometries, nothing terribly exciting. For some of the nuclides of interest, and there's some dozens of them, we could use some improved nuclide data. But apart from that, the future, as I see, is in the introduction of gamma-gamma coincidence systems at our monitoring stations, and of course also in the laboratories. Now, the beauty is, of course, they can also operate in a singles mode. So we have, we can still operate our network, even if one of those detectors fails. But in coincidence mode, it promises improved sensitivity for, of course, for some of the radionuclides. So the picture there is a test system we have here in Vienna, just a few levels above my head. And we use that to look at some of the benefits we could achieve by using a coincidence counting. There are different ways to look at it. It's one, of course, we have the stations and we have the labs. Laboratories can count for longer. Stations have a higher, the samples have all kinds of radon daughters on it, makes it more complicated. But in essence, if we look at a typical stations, how we operate currently with a single germanium detector compared to coincidence counting, and the right-hand side, you can see for the example of lanthanum-140, you can achieve factor of 10 or so improvement. That's quite significant. And if we look at an example when folks can work through scenarios, for example, a release from a hypothetical test site in the DPRK, one of the closest monitoring stations that we have is in Takasaki, Japan. It's about 1,000 kilometers. And you can work through this exercise where if you have a release where we may actually not detect the release with the current monitoring station, but we would in fact not only detect the release with a coincidence system, but would also be able to determine the zero time. But there are challenges moving to gamma-gamma coincidence systems, not only to do with data transfer, etc., but also the validation of the calibrations and the models we use. Of course, we can we have means to with test samples to validate. But one thing we've found that's the nuclide data format that's currently available makes it a bit cumbersome to work through this exercise to set up the cascade summing effects in the calculations to determine the correct activities. So that could be an area for discussion, either in this forum or ICRM maybe. And of course, you could go to nuclide-specific calibrations, but then we would need a large number of certified reference sources with all the different, at least a number of different nuclides, which is currently not available. Moving to noble gas monitoring, there are currently our network is set up to monitor for xenon. The xenon isotopes, there's 4 of them that have reasonably high fission yields and suitable half-lives. But we have increasingly issues with the atmospheric background from medical isotope production facilities, but also reactors. And data seems to indicate that BIPM is operating a detector that every few months releases quite a large amount of xenon, which would prove a challenge then for us to detect a nuclide, if that coincides in time. So one thing for the future that could be of interest is to include argon-37 in our network. There's very little background in the atmosphere. It's not produced in reactors, or not much. But it's quite difficult to measure with the sensitivities that we require. For the noble gas systems, we have the challenges that we have gaseous sources. So if you want to introduce them into a system, there's an unknown transfer efficiency. We have short half-lives, but a global network. There's some systems work with gamma spectrometry, so there, of course, that's easy to calibrate. Otherwise, we work with xenon calibration sources and use an absolute calibration process. Then we have a QC program for our noble gas monitoring network, where we, to ensure the system is operating correctly, we have we have a reference lab that produces xenon reference samples for us that can be with 2, 3, 4 isotopes. This is certified, although based on gamma spectrometry only. But because we do not know the transfer efficiency when injecting these spikes from these certified values, we can really only use the ratios. In the system, the samples are measured. We get the activity concentrations and the xenon volume. And then the system set up that we can transfer these samples to one of our IMS laboratories, where they can remeasure the sample, and then we can compare the activity concentrations and the xenon volume. The only limitation, again, is the half-lives. So at the laboratories, we usually only measure xenon 133 and 131M. For noble gas, as well as for particulates, we operate proficiency test programs. And for xenon, again, the challenge is the short half-lives. There we have our reference lab, is the Idaho National Lab in the US. The PTE samples for the IMS labs have activities in around becquerel range, whereas the actual atmospheric samples are more millibecquerel. Now, if you want to do a comparison with the primary measurement, we need about 100 kilobecquerel. So there's a bit of a challenge to do this, but we went through an exercise recently where the reference lab produced samples with they were based on the same gas sample that was split, so that the activity concentrations were the same, only the volume was larger. And then our laboratories had the option to wait for months, and then they would have activity values that were within their measurement range. In this case, we had an exercise with LNHB, and oops. Here are the results. That was quite reassuring. So the INL, so on the left-hand side here is a comparison with the normalized value for LNHB value. The INL value is based on gamma spectrometry. Error bars here are 1s, so this is excellent agreement within 2%. And then on the right-hand side, the two left values are the same, just different scale. And then we have the 4 labs which provided values in this exercise from us, from our labs, with, of course, much larger uncertainties. But overall, the agreement is quite good, which is really important for us. So challenges for noble gas monitoring, we have currently traceability through gamma spectrometry only. But of course, there's a program from the NMIs to set up again noble gas, gas counting capabilities. That's really reassuring. But also not only from NMIs, but also for other laboratories, there's only a very small number of laboratories with xenon measurement capabilities at the low level range. We have to bridge the gap between the environmental levels at millibecquerels to the 100 kilobecquerels, 10 to the 8 or something ratio in between. Because of the complexity of this, there's only a limited number of measurements that can be undertaken. This is slow progress. In addition, as I mentioned before, we have a varying and increasing atmospheric background on xenon, which makes the interpretation difficult. There is an option to move to argon, but that involves still involves some technical development. So in summary, we operate a worldwide network of radionuclide monitoring stations and laboratories, where we measure trace levels of radionuclides and aerosols and novel gases. The radionuclides of interest are short-lived, where short here means in the days to years. We're sitting in between different communities. We have some overlap with medical isotopes, but not completely. We have little bit of overlap, but not much, with other environmental monitoring programs. And from the what we're looking for is from the community would be improved nuclear decay data, and in some cases a discussion on the nuclear data format to, if necessary, we might be wrong, to improve our process for setting up gamma-gamma coincidence counting. Primary gas measurements, of course, and the key comparisons that are coming up are very interesting for us. Reference materials for xenon and the future potentially argon. And with that, thank you very much. And there's time for questions. I'm happy to answer those.
Sometime. I'm a radio pharmacist by training, and for 4 years I worked in the clinic in nuclear medicine in South Africa, where I was born. And in thereafter I came to Belgium, and now I work at KU as a preclinical and also radiochemist. So I develop try to develop new theranostics for cancer. And this is why I have such a passion for this topic. And I was really very glad to be invited. Yes, we work with small amounts, but we even have issues with higher amounts, and this is why I'm going to cover one issue. I said I will talk about multiple issues, but I'm actually just going to talk about the dose calibrator in the nuclear medicine clinic. We are talking now about the implementation of more exotic radionuclides in the nuclear medicine clinic. And I feel at the moment we have a very false sense of security. As a radiopharmacist by training, we get this book with every dose calibrator. There's these codes, and I brought it with you. I guess I'm one of the only speakers bringing a book. And then there is all of these disclaimers in here, but actually nobody in the clinic believes these disclaimers, and they believe the code that you put in is the one that will work, or the one you even put in another dose calibrator, you go somewhere else, you know this code, you put it in. And there is really a big problem with how we measure radioactivity in the nuclear medicine clinic. We are entering an era where therapy with radiopharmaceuticals is gaining a lot of momentum. And these are quite dangerous because we are labeling molecules with alpha and beta minus emitters and injecting them directly into patients. And I think this is really a big issue. And as a radiopharmacist, a radiochemist, I come here today to say there is a gap between physicists and physics and radiopharmacy. And I don't know how to solve it, but it gives me sleepless nights about the future of me and others that are starting on this journey to implement exotic radionuclides. And the pharmaceutical companies are pushing this, but they're not taking the responsibility. So there is 3 issues, of course: quality control programs that is run on a daily basis in the clinics, the measurement of the containers used, because these codes are done by very specific ampules, but we are using diabetic syringes for preclinical or radiochemistry. We are using 10 ml syringes for patients. We are using vials. We are using multiple types of vehicles or vessels to measure, and then, of course, the calibration factor selected. So there is the manual that I showed you, and this is a dose calibrator. You know maybe better than me how it works, but it's the ionization created by the radiation that is measured, and then this charge difference is converted. And then also you put in the code to say which activity you're working with. And I think this is a really great and helpful instrument if it's used by people that know how to do it. But again, the technologists, the pharmacists, all these people, the chemists, we are not physicists. So how do we bridge this gap? And these days, the alpha emitters is coming forward. So talking about small amounts, we want to measure for patients as much as 6 megabecquerel of actinium, or you know, it's an alpha emitter, so you are measuring the daughters. We have lead to 12 coming, where when you have the lead from the generator, you have to wait at least 4 hours before you're able to measure, or the measurements change because of all of the emissions. I was at an astatine conference just the last 2 days in Nantes, and they don't even know how to really accurately measure astatine, but yet we are doing clinical trials in humans. And I would always ask, as a pharmacist, would I inject this into myself or my family? Will I be so inaccurate? Is this okay? And for me, this is really not great. So quality control. In many institutions in controlled countries, this is done really well, but there is also many places where just the basic daily QC is not being done. And then as a researcher, in the preclinical setting or radiochemistry setting, often this is neglected completely. Yeah, so there is a paper here about the accuracy of dose calibrators for gallium. Gallium is a diagnostic radionuclide as we use it for PET. I was looking for papers where they describe just how good quality control is being done in the clinic, and I failed to find some, but I know I've read on this before. This is really one of the basic things not often being done great. The measurement containers being used in geometry is a really big issue, and now Terbium-161 is coming forward, and it's really being pushed on the market, and it's being shipped to all over the world. So the reproducibility of placement and where and what vials we use really messes up our accuracy. And I just here having to show some of the work done at EZ11 by the physicists that know their stuff. And they saw that maybe the 10 milliliter syringe has quite a big deviation. And this is the one we used to inject the patients with. And this is a facility that is a really state-of-the-art place. They care about the physics, but terbium-161 is used in a whole lot of places. And then even for preclinical, which I do, I got 3D printed discs to put in my dose calibrators, and then we can develop for our dose calibrator and the geometry of the specific vessels I use, we can develop specific calibration factors for our dose calibrator, but this is not being done. So, yeah, so radionuclides particularly susceptible to the geometry effects is beta emitters, but also the alpha emitters. These are really a big issue and we have to look at it, and it's coming. Then the final thing is the calibration factor selected. And recently I work on rhenium-188. Do I still have time? She's not looking, I'm sorry. I was like, I'm trying to send a serious message in 10 minutes. I can talk about this for days. Recently I work with rhenium-188 and I thought, oh, this is not a new exotic isotope. We know about rhenium-188 for a very long time. And I asked SCK to come to me. They asked quite a bit of money, but it was worth it, to bring their Fidelis machine for a secondary standard. Yeah, and we did this whole exercise. I tried my best to understand everything that's going on as a pharmacist. And so in the end, we actually realized that there is up to a 15% deviation of rhenium as measured by this very good secondary standard machine compared to the factor that is printed in this book. So this scared me. And of course, I think it's also not only therapeutics, but therapeutics is really important because you can underdose the patient and then you can get, you could have done a lot more for the patient. The cancer could have cleared much better. Or you can overtreat them and have maybe subclinical kidney damage and these things, or even clinical kidney damage. And this is just from a reference, quite an old one, where they just said, yeah, radiopharmacy is imprecise, and it may be possible to improve. Now, I challenge us all to think about how we can definitely improve this, because we are talking about human beings here, and also they really need the therapy. It's amazing what we can do, but we have to do it accurately. And then to just conclude why accuracy matters. So for instance, as a radiochemist, it's very important for me to be repeatable with my production protocols. So often when you interchange between facilities, your methods, it suddenly doesn't work. And maybe it's just because we are inaccurate with the amount of radioactivity we measure and add to our production procedures. There is a huge variability of data in the research and development space. Often we are told, why are we doing animal research? Why are we doing these things? And it never maybe translates that well into the clinic, but we are measuring very small amounts in the animal studies. So this is one of the major reasons, in my opinion, why we cannot really translate everything we see in animals. And then I think the last point just goes without any further talk about it. There is a big risk associated with inaccurate patient dosing. And then just to see, I go all over the world. I travel a lot in Africa, in the Middle East. The dose calibrator is always there and the manual is always there and people are always using the code. So it is not a problem for just for us that do these exotic radionuclides. It is all over the world. And it is often in these vulnerable communities, I think even bigger because the dose calibrators is older. They're not maintained properly, and the pharmaceutical companies ship terbium-161 and actinium even to these places. So I want to thank you for your attention and also acknowledge my colleagues and specifically Prof. Christoph Potter at KU Leuven, because we talk a lot over coffee about this problem. Thank you so much.
Good afternoon, everyone. Well, as Lisa said, my name is Ivana Vukanac, and I'm coming from International Atomic Energy Agency. And today I will give you one general presentation about low activity concentration measurements, and we will touch a bit sensitivity of radio analytical methods and the role of reference material in analytical process. So summary of my presentation. First, I will give you a short introduction about the laboratory that I'm coming from. Then we will go through performance characteristics of analytical methods and methods that are used for activity concentration measurements. We will have some slides about usage of reference material in analytical process. So, Turk is one of 4 environmental laboratories in the agency. It has a long tradition. It was founded in 1960s, and since then it has been growing in its activities, expertise, quality of performed work, and since 2022, the name of the lab is Terrestrial Environmental Radiochemistry Laboratory, and we are part of Division of Physical and Chemical Sciences of the Nuclear Science and Application Department. So our mission is key task of the Turk laboratory is to provide expertise, training, and support to the member states dealing with radioactive, industrial, and any other pollution, and to assist member states in preparing for the emergencies. Laboratory supports member states' laboratories in improving their measurement capabilities, in environmental monitoring, and in providing reliable analytical results. And we do that by providing reference materials and by organizing worldwide proficiency testing exercise once per year. For that purpose, we have infrastructure, not only for measurements, but also for reference material production, their storage and distribution. So to fulfill our mission, we have, of course, equipment, and in our laboratory, we can perform stable isotope analysis, trace element analysis, and of course, radionuclide analysis. And for that purpose, we have well-equipped gamma ray spectrometry laboratory. We have alpha particle detection system. We have liquid scintillation counters, gas flow proportional counters, and for sample preparation, one nice radiochemistry laboratory. We are speaking, well, this workshop is focusing on trace level, on low concentration measurements. So I would like, to say what is low and what is high. The answer to this question is not that straightforward. It strongly depends on the perspective in which field you are in. We have application of radioactive material in many fields. And for example, if we speak about industrial application, for example, sterilization, we are talking about activities 10 to 15, for example, becquerel of cobalt-60, or in medical application, high activities like 10 to 7 becquerel in diagnostic and 10 to 11 becquerel in radiotherapeutical application of various radionuclides. But if we are in environmental measurements, then we are speaking about several tens of becquerel per kilogram of radionuclides, natural ones, members of uranium and thorium chain, and about several hundreds of becquerel per kilogram of potassium-40 if we speak about soil. If we, for example, speak about water, activities are even less. So what analytical method we are going to use to measure strongly depends, not strongly, but depends on the activity that we expect that we are going to measure, and of course, radionuclides that we want to measure, and the method we choose based on performance characteristics. Here on this slide, you have very useful, nice document, EURAHEM guide, which lists performance characteristics that needs to be assessed prior two measurements to see if the method that we choose is fit for the intended use. So in next few slides, I will go through performance characteristics according to this EuroHEM guide. We have selectivity. Well, I will not read the definitions, you can find it in the documents, but if we speak about radioactivity measurements, then selectivity means that we can detect and quantify certain radionuclides in presence of others. Next one, calibration function. It is very important. Well, in laboratories dealing with environmental samples, usually we use relative methods. So detection system needs to be calibrated and calibration function needs to be established. Then we have limit of detection. In radioactivity measurement, a limit of detection is the smallest amount of radioactivity that can be distinguished from the background. Limit of quantification, again, is a smallest amount of the activity that can be detected and quantified with acceptable uncertainties and accuracy. Then we have working range. Says for itself, it's interval over which the method provides results with an acceptable uncertainty. Analytical sensitivity. This is smallest amount of an analyte that can be detected by applied method. To assess this analytical sensitivity, it can be useful to form a response curve, and the slope of this, it's usually linear calibration curve, linear response curve. Slope of this curve will tell us something about analytical sensitivity. The steeper the slope, better sensitivity. Then we have trueness, precision, and uncertainty. Three performance characteristics that are used according to EuroHEM guide to assess quality of the measurement of the results. Trueness is usually checked by measuring by measurement of some reference quantity value, and on that way, this way we can assess bias, possible bias of the method. Then if we repeat measurements of the same sample under the repeatability under the specified conditions, then we can see how our results are spreading, and from that we can assess standard deviation of measurement result. Third third performance in this set is the uncertainty of measurements, and this will give us a range where we are certain with a certain probability that true value of a measurand is. And we usually evaluate a combined uncertainty of measurements using one of these two approaches. First one, bottom up. Well, personally, I'm always doing like that. So first we need to identify all contributors to the measurement uncertainty. Then we need to quantify them, and sorry. And at the end, we need to combine these contributors to a final uncertainty measurement of the results. In a top-down approach, we don't have complete understanding of all influencing parameters required, and the value of the uncertainty needs to be estimated based on, let's say, results obtained during method validation, or after analyzing results of participating in proficiency testing or in interlaboratory comparisons. So, as I said before, methods that are used for activity concentration measurements are chosen. Its selection is based on the capabilities and limitations of the various techniques, including selectivity, sensitivity, range of measurement, is the technique is complex, and do we have all the resources needed? In general, there are two approaches for quantifying the radionuclide content in the material. Radiometric measurements, where we detect characteristic radiation, and mass measurements, where we simply count number of atoms of a certain element. So for these mass measurements, we heard about this earlier today. Measurement is isotope amount ratio or a mass fraction of certain element in solid or liquid material. These measurements are highly sensitive, and we can detect trace levels like micrograms per grams in solid materials, or ultra trace levels like nanogram per gram in solid materials. These measurements are most commonly used for heavy metals, let's say thorium and uranium, and the application of this method implies, again, rather complicated sample preparation before measurements. What is different than radiometric measurements also as a result, in the mass measurements, we don't have becquerel or any other unit of specific activity concentration. We have ppbs, ppms, and after measurements, after obtaining results, some conversion from these units needs to be done, and it is number is different for each radionuclide. Here you have an example for uranium-238 and thorium-232. About radiometric measurements, so there is a lot of radiometric techniques that you can use for measurements, and I've put in this presentation the ones that we have established at Turk laboratory. So these are alpha particle spectrometry, beta counting, liquid scintillation counting, and gamma ray spectrometry. For first three, we need to do radiochemical procedure, meaning that we need to isolate from the matrix of the sample radionuclide or radionuclides of interest, and we need to do that with high yield and with high purity. First of these is alpha particle spectrometry. This is one very nice method, and it would be in environmental labs key method for identifying, of course, alpha emitters, usually used for actinide measurements. It is very suitable for measuring very low activities, and we can reach activity concentration lower than millibecquerel per kilogram. But this method is very well demanding. It requires complex sample preparation. It lasts a long time. So these are the drawbacks for this method. Next one, beta counting. Again, essential is radiochemical separation, and after source preparation with different techniques, usually it is measured by gas flow proportional counter, and this technique is used for low-level analysis of beta-emitting radionuclides. Reachable concentration are becquerels per kilogram, order of becquerels per kilogram of, for example, lead-210 or strontium-90. Then we have liquid scintillation counter, very similar. Measurements are different, sources are different, and equipment, of course. This method is used again for low-level analysis, not only for beta-emitting radionuclides, And again, lowest concentration that we can reach is becquerel per kilogram of sample. And at the end, I put gamma ray spectrometry. Well, not at the end, it's the different, the previous three, but it is, I would say, like a queen of environmental laboratories, because with this method, you can detect multiple radionuclides at the same time. Sample preparation is not that complicated. Usually it's just mechanical preparation of your sample. And you can do detection, identification, and quantification if you have nicely calibrated measurement equipment. This is a nice thing because in measurement of environmental samples like soils or I don't know, industrial waste products, you can find, I think the number is 36 different radionuclides, and you can detect all radionuclides from that list in one spectrum if they emit detectable gamma radiation. Levels of activity in environmental measurements are about becquerel per kilogram up to hundreds of becquerel per kilogram. We spoke earlier, I spoke earlier about this Eurochem guide, and the practitioners in the field of radiometric measurements use ISO guide 11929 for determination of the characteristic limits for measurements of ionizing radiation. This guide is incorporated also in some softwares for calculation of this detection limit. So in this ISO terminology, limit of detection would be a decision threshold, and it will give you an answer to a question: is the net peak count in the region of interest significantly higher than the background? Detection limit would be like a limit of quantification, giving you the answer on question, what is the lowest count in the region of interest that I can detect above the background at a certain confidence level. But limit that is most often used by practitioner is minimum detectable activity. It's like a detection limit, but in units of activity. And if we cannot detect a certain radionuclide in a measured sample, then we say it's less than MDA or MDC like concentration. And we can improve this value if we have lower background and if we have good detector with nice efficiency and resolution, if we prolong time of measurement, and if we optimize detector to sample geometry, making the solid angle and efficiency as maximum as it can for this specific measurement. Now I will tell you something about reference material usage. I hope that many of you are familiar with this ISO 17025, which gives general requirements for the competencies of testing and calibration laboratories. Clauses 6 and 7 of this standard gives resource and process requirements, so equipment and methodology used. And here in details, you can find how reference material can be used in analytical process. In summary, reference material can be used in all stages of analytical process. They can be used for calibration, certified reference material. Then they can be used for quality assurance and quality control activities, which includes both method verification and method validation. This slide is just an example how it works with the gamma spectrometer. Earlier today, we spoke about traceability of measurements. If we use certified reference material for calibration, then we take traceability of this reference material in a chain of the traceability of our measurement results. So if we can go in more details here if it is needed. I said at the beginning that Turk Laboratory is a producer of reference material. We are accredited by ISO 17034 since 2022. And our radionuclide reference material are traceable for becquerels to national standard of activity of radionuclide through efficiency calibration of our gamma spectrometers, and for kilogram to a national standard through regularly calibrated balances. We have available approximately 100 reference materials, and we distribute about 2,000 units of reference material to over 600 laboratories all over the world, approximately 80 countries per year. And here in the conclusion, I just listed something that I wanted to leave with you after this workshop. So first, as I said, low activity concentration measurements are, let's say like that, and relative issue. We need to see low compared to what. Then purpose of the measurement and expected levels of activities that we are going to measure need to be assessed prior to a measurement. Also, performance characteristics of an analytical method needs to be assessed, and chosen method needs to be fit for the intended use. As I said, reference material can help us in our analytical procedure. And I would like to add that the quality of obtained results directly depends on the quality of used reference material. And at the end, well, how things works in environmental laboratory, I think that it is the same all over the world. So when you have your unknown sample for which you need to determine content of radionuclides, first you do gamma ray spectrometry, and if it is necessary, then you take some hard artillery like alpha particle spectrometry or mass spectrometry to reach lower levels of activity concentration. That's it. Thank you very much for your attention.
So today I would like to talk about the digital transformation and radionuclide metrology. So it will include 3 sections. First, it's a very short introduction about the relationship between the digital transformation and the radionuclide metrology. And after that, I will separate to discuss, to introduce. So for my understanding, the digital transformation and the metrology should cover 2 dimensions. The first is the digital transformation for metrology, which is to apply the digital technologies to modernize the metrology itself, and for nuclide metrology, of course. And so this will cover the measurement instrument, the process, or even the service, the digital service. And the second dimension will be the metrology for digital transformation. So this is to use the application of the metrology to entrust to ensure the trust and the reliability for the digital technologies. So now I would like to move to the first. It's about the digital transformation for radionuclide metrology. So as we already talked about that, it's to use the digital technology to modernize the metrology itself. So what kind of the digital technology we can use? I just searched from the digital technology. So a few years ago, when I asked some questions from online, I cannot have many very high quality results. But now with the AI, the large language model, it's more easier to find the very good result. This is kind of the digital technology. So for example, in our fields, what we use very often is the digital acquisition. And of course, we can use the digital twins, but yeah, we have some discussions. Maybe it's in our fields, it's more on the simulation. And also in most of the discussions, the artificial intelligence or machine learning can be included in the digital transformation. And also we can use cloud computing, the edge computing, and Internet of Things, many. So many technologies. And I will just give some examples for some of the technology which will be more related to our fields. For example, the digital acquisition. So in our fields, we use this from very early years, and we use the digital acquisition and to acquiring the data. It's very common, but after that, we will save it in some digital data format. Now, very common format is the list mode format. And based on this kind of the format, we can do the algorithm-based analysis for the signal processing and for the activity estimation. So it will be the software-defined instrument after. And so in this application, by the digital acquisition, we combine both the digital acquisition and the software-based analysis. So this is some history in our field that in 1996, some colleagues, the BIPM and other colleagues from Einstein already introduced the concept of the digital coincidence counting. So, and also our colleagues, John Kisley from NPL, Tisu Pan from South Korea, they're doing many studies in these fields after, and I just show some examples. They're doing this digital acquisition by custom-built digital acquisition device. So not so many institutes following at that time because we need some skills for the development of the digital acquisition device. But now it's more easier to find the commercial one. So we can use the commercial one to do the digital acquisition. For example, the digitizer is very common used the device. So we're also doing something on this and just some examples. So we can use this digital acquisition for our single process acquisition for the beta gamma coincidence counting. So this is to use the develop the software defined instrument of the beta gamma coincidence counting technique. And also another example, it's very similar, but it's more complex, is to use the TDCR gamma detection setup. And we can develop some method like the coincidence counting or even other method like the selective sampling method. So in this application, we can define the function of the instrument by software. So it's very flexible and easy to develop something. And many other applications in our fields, like the TDCR-G detection system from Platon and the TDCR gamma system from PDB, from the TDCR from ENEA, from LNGB, many applications. I just list some of the examples. Okay, now I would like to talk about the digital twins. As I said that we have many discussions. So in our field, maybe from the definition of the digital twin, it's a dynamic replicate of the physical object. So in most of our applications, maybe we are not the true digital twins, but we are more like the virtual measurement instrument based on the simulation, especially the Monte Carlo simulation for radiation transport. We have so many well-developed codes to do the simulation. And for this application, this technology, the virtual measurement instrument, I thought about 3 different applications that we can use the Monte Carlo simulation to, we can build the virtual instrument to predict the efficiency like the gamma spectrometry, to predict the spectrum and many other parameters. This is what we do very often. So we can see how well the model will fit the experiment. So recently, also we think about some other applications by this technology to build the virtual instrument to generate the digital data set, which will be very useful for the validations of the algorithm. So we generate the model and we create the Monte Carlo model and to generate the data into the digital format, which we can have the true activity, the true reference. And it's a very good way to validate our algorithm developed by different laboratory. And the third one I thought about is the metrology for simulation. So this idea I learned from the PDB's publication, but I think it's quite interesting for us, and we need to think about it's the metrology for simulation. We will think about two things that we can, when we have the Monte Carlo the models. Previously, we only think about how good the model can fit the experiment. But even the model is not good, we can think about we can calibrate by the models by experiment. Even the model is not good, we can estimate, we can evaluate the uncertainty of this model. And after that, we can use the calibrated model to do some predictions. And that means it's the simulation for calibration. So in this way, maybe we can build the digital traceability of the simulation. And based on the previous two technology like the digital acquisition and the simulation, in our fields, we are doing something, it's for the algorithm comparison based on the digital data set. So this comparison is a way to validate the algorithm, which is a little bit different from the conventional comparison. The conventional comparison is based on the measurement of the distributed radioactive solutions. But now we would like to analyze the distributed least mode data set. And the data set can be coming from the experiment, use the digital acquisition, or from the simulation. And we can have the digital data set and to analyze by different algorithm driving code to compare the performance and to see how to validate the algorithm in very good in a very good way. So for the algorithm comparison based on, I give some examples that algorithm comparison based on the experimental digital data set, we're doing some comparisons, bilateral comparisons, use the data from PDB and do some comparison with different code, the code from PDB, the code from NIM. And based on this analysis, we found some advantage. So for the traditional comparison, it's always affected by the statistic. But if we use the same data set, it will overcome the problem of the statistic because we have the similar statistic. So that means we will only compare the difference between the implementation of the algorithm. So in this comparison, we found that the maximum relative deviation for different analysis is less than 0.03%. It's about 10 times lower than traditional uncertainty. And another example is about the algorithm comparison based on the simulated digital data. So as we say that we can generate the data, and after that we can analyze the data by different software, different algorithm, and it's a rigorous validation of the algorithm. So as you can see that for this validation, we can find the relative difference is in the order of 0.002% in this level. So it's very, it's very precise validation of the algorithm. Okay, so let's move to another technology. It's about the cloud computation. So what we, as you can see that in the first 3 days, today, we use some kind of the tools like the SharePoint. So the SharePoint is kind of the cloud. It's a collaboration cloud. We can do the real-time co-work on the documents, to share the data, to exchange data. So it's this is one of the tools we can use, but also we are thinking about to use some compute cloud for the training of the AI, training of the machine learning, or we need to exchange the big data. Since we have a lot of data in big size, it's not so easy to transfer. So there are many different clouds, and we use. It gives us a lot of benefits. And now I would like to move to the artificial intelligence and the machine learning. So as you know that there's so popular fields in nowadays, and so for the artificial, I'm more talking about the machine learning. So as you can see that we have many different method for the machine learning, like the regression, the classification, the clustering, or the dimension reduction is included in different applications. And also we can have the generative AI, the deep learning, many things. There are some, also I put some applications I found in the literature that in our fields, many people do to use the machine learning method to do something like the parameter optimization, the activity estimation, the radionuclide identification, something like this, discrimination of the signals. So there are some examples, very simple examples, but we found some benefits that, so when we want to tell the difference from the background and the real event, it's not so easy for traditional way. Usually we tell the difference by the pulse shape discrimination. But by the traditional way, it's not so efficient. And so by very simple machine learning, like the supported vector machine, the very simple one, we can have a very good performance. We can have we can significantly reduce the background, but still keeping the detection efficiency. And this is the same example that we can tell the difference between the background and the real event. And so on the right, it's the performance of this. And another example, it's also a very simple example. It's based on the unsupervised machine learning. It's just to compare the traditional way to doing the alpha beta discrimination and by the machine learning method to discriminate the alpha beta. So as you can see on the left, we place several different methods, like the very traditional one, the PST method, the pulse shaping discrimination method. It's very often that we cannot totally separate, or you adjust the parameters, but it's still the performance is not good enough. But just by the machine learning, like the cluster, use some very recently cluster method, clustering method. So we can see it's very easy to separate alpha beta, totally separate. And after that, when we go back to the spectrum, we can see the spectrum of the alpha, the spectrum of the beta. So it's very easy to do this work, but in traditional way, it's not so easy. And also some other applications like use the CNN. It's quite popular to doing some radionuclide identification. So the examples is with different counts in the peak. So you can see the performance of this. In most of the cases, the performance is quite good, that even the counts in the peak is very low, it still have a very good performance to identify the nuclide. And also when the singular to noise ratio are not so good, it can also have a very good performance. Okay, I just give some examples, but not all the examples for different digital technology. And now I would like to introduce the radionuclide metrology for digital transformation. So the metrology for digital transformation is to use the metrology to ensure the trust and the reliability for the digital technologies across the industrials. So what we can do, I search for some literatures, materials, and to learn what we can do for metrology. For example, now we have many digital metrology surveys, like the most popular one is the DCC. Most of people talking about the DCC, digital calibration certificates. And also now the digital reference material certificate is similar, but yeah, it's also popular now. And the digital comparison reports, the similar structure. And also we can do in the digital standards like the digital SI, the digital reference data, the digital semantics, like the digital VIM, and metrology specific semantic models. Or we can generate the traceable reference data for the AI machine learning validation or trainings, and to generate the simulated data by digital twins, and also some other applications like the fair data, the metadata. So for most of this application, I found that in the framework of the CIPM, so we have a forum on the metrology and the digitalization. So in this forum, there are many groups, the task group, working group, cover most of the application for the metrology, in the dimension of the metrology for digital transformation. So I think this is more about how to make some rules, make some definitions, and make some surveys to use the metrology method to provide the service to industrial. And so what's popular, just some examples that the DCC is developed quite well. And also recently, there's one application in radionuclide metrology. It's a DCC for radioactivity by PDB. And the digital comparison report is developed in our tasking group. So it's developed mainly by Holman column from BIPM, but this digital comparison report method was structure was it's the machine readable version of the comparison report, and it's reviewed by the CCRI digital transformation tasking group. So just to show the example of this. And also we have the digitalization of the service categories. So all of these are digital service. We have some applications in our fields for radionuclide metrology. And very recently, we also have the digital identification of the nuclear data. So now we have the DOC for the reference of the nuclear data. Okay, that's all. Thank you for your attention.
Yes, I will show you some news about our reference system and comparisons. Reference instrument is an international reference system, the SIR, for gamma ray emitting radionuclides. It's based on an ionization chamber at the BIPM, pressurized ionization chamber. The participants, the NMIs, send us an activity standard solution in normalized Gleis sample that we measure in this chamber. And we calculate the result, which is an equivalent activity. It's, in fact, the activity in the ampoule as measured by the NMI in its laboratory, divided by the current that we measure relative to currents of iridium source. So it's what we call an equivalent activity. And in fact, it's more or less equivalent to the inverse of a calibration factor of that instrument, except that it's here you have the radium sources coming in. It's a simple on-demand service, and it's run by the BIPM since 1976. It's based on the high stability of the ion chamber. So you see here the radium measurement results, the stability check, and also the ratio between the different radium sources that we measure. So since 1976, so it's very quite stable. And that's thanks to that, we can compare equivalent activities measured recently with equivalent activities measured 30 years ago. And because the instrument is stable, we can compare them. So thanks to that, we accumulated about 80 comparison results from 30 different participants and 73 radionuclides, from which we have 60 KCRVs, which are special means of selecting the equivalent results over the time. Some are sometimes very old because we don't have many measurements for that nuclide. However, there are some drawbacks or this limitation of the SIR. First is because it's almost 50 years old. And who knows when it may fail tomorrow or not, or leak. And so we recently published a methodology to transfer the KCRV to a backup ionization chamber that we have together, and we monitor for 1976 also. Of course, this transfer would increase the uncertainty of the KCRVs, but we have shown that it would have a negligible effect on the degrees of equivalence, so that's fine. The problem is that also the backup ion chamber is 50 years old, so we just postponed the problem, so I will see. We will see that later. Another issue is that we are using the same electrometers since 2008 with a homemade Townsend balance approach, and if it would break down, it would not be easy to repair, and also there is a limited choice of electrometers on the market. So we would like also to have a new current measurement system. So we did both at the same time. So we have 2 new ionization chambers. So you can see here in 2 separate shieldings, and using a new current measurement technology. And we had Dr. Tanaka from NMIJ who came and optimized the system and validated. So we use an ultra low noise current amplifier, the ULCA. It's made by a company, Manicon, close to PTB. And also you need a Keysight digital voltmeter. This system has a particularity to have a very high accuracy, stability on long term, and linearity versus a current. So you have the ion chamber, and you connect your output in this ULCA. For example, 100 picoamps would correspond to 0.5 volts. So because the gain of this ULCA is 5 × -9 volt per ampere. And then you have the voltmeter to measure that. So using a Keysight, but we also tested a Keysight digital voltmeter. The good news is that after several months of work of Dr. Tanaka, we finally obtained a current precision similar to the present SIR. So you see 200 picoamps, we have less than 10 to -4 type A uncertainty, and a little bit worse, but still very good for 2 picoamps. So we have less than 0.1%. So it's a good news, and that means that when we will have some time, we will try to test this new system on the old SIR and see if we can replace the old electrometers. Then we also try back-to-back connection. That's what how they call it at NPL, where they developed, let's say, this method. They tried the first time. So you connect the 2 ion chambers polarized with opposite polarity to subtract the background. So you polarize, let's say, positively the left on the left and the one on the right with the negative, and you put the ampoule in one and measure the background in the other. And so if you just connect the 2 cables, the output cables, you just subtract the background continuously. And if the background is fluctuating over time, it's taken into account immediately. So it is very good idea, and it seems to work pretty well, because of course the chambers are identical and have been matched a little bit by the manufacturer in efficiency, but and thanks to that we have an apparent background measured lower than 1 femtoamp. And there is also an offset from the ULCA amplifier of 15 femtoamp. But it's very stable. It's not a background which is more random. There is just an electronic offset. And so we were enabled recently to measure 30 femtoamp over 5 days, and we ended up of a 0.1% uncertainty. So that's very encouraging. And now, so it seems that we could make comparison at current levels 15 times lower than for the present SIR. So, and well, still a few things to improve, but it's in progress. So that means that the future capabilities of the SIR with the ULCA-1 and back-to-back connection is a probable expansion of the SIR capabilities to lower activities, and that would help for rare and expensive radionuclides. Also maybe help for the transport. But lower activity is not at the level of what we have heard before, but still, it could help. Also, the chamber has a larger well than the present SIR, so that would allow to compare samples larger than just 5 mL ampoules and open the doors for new applications, so measuring gas containers or liquid scintillation vials or even reference material, maybe with high activity, of course, not the lower one. Other limitations of the SIR, of course, it's measuring gamma rays. And if you have an emitter like americium or cadmium-9 with low energy photon, you have few charges to measure, you have a larger uncertainty also from the glass thickness of your ampoule. And if it's a pure beta and alpha emitter, you just don't measure anything. So you need something else for this kind of neutralize. And that's what we call the extension of the SIR, the ESIR. Based on liquid scintillations for beta, electron capture, and alpha radionuclides. So this is a scheme of what it is. So you put at the middle of the instrument a liquid scintillation source. That means that you mix the radioactive solution with this cocktail, scintillation cocktail, and it emits light, and then you detect the light with photomultipliers. This system is based on the TDCR models. It's a kind of primary measurement model and a specific comparison indicator minimizing dependence on detection efficiency changes over time or over the sample. It has a digital electronics, and here it has been validated, with a pilot comparison. And this is the list of radionuclides agreed by the CCRI to compare, and I opened the service recently. We also added recently tritium and chlorine 36 has been agreed this week. We have received 3 first samples this year that have been measured, but no result yet. So my colleague Romain and his colleagues made enhance of automation in the process with a digital notebook, a Python code, Git control, etc. He also tested the cocktail versatility. So depending on which liquid scintillation cocktail you use, and you could see on the graph that you have the results in agreement. And also electronic redundancy and versatility. So if the LabZ digital electronics would break down, you can replace by another one. And because on the right, you see that the results are the same. The perspective is that to measure more and more ampoules, and hopefully several ones from the same nuclide, so you can compare, but for the moment it's not the case. And an extension to alpha emitters. So we have new capabilities in source preparation, thanks to our new staff member, Tiffen Choteau. And so there will be new source preparation facility for alpha emitters in particular. And so we hope to measure, be able to prepare the cocktail and measure the americium-245 from NRC in the next year or so. Finally, another limitation is because of the decay during shipment. So when you receive a sample from overseas, the sample decays during the transportations, and so you can't measure it. So the overseas laboratories cannot participate for short-lived radionuclides. And even within the European Union, it's a little bit challenging if it's shorter than 2 days or let's say 1 day. But it's of crucial importance for SI traceability in medical applications because they like to use short-lived radionuclides. So with transport, at the request of the CCRI, we developed a transfer instrument. It is traveling. It's based on the well-type sodium iodide counting. Well, it doesn't matter details, but we are just making counting above a low energy threshold and count everything and make some corrections. It has a high reproducibility because each time we dismount it, remount it in another lab, and also stability. is traveling to the NMI's DIs for comparing short-lived radionuclides. And this is an example for technetium-99M, the widest radionuclide used for imaging, 6 hours half-life. So on the left, you have the results from NDSIR for laboratories close to BIPM. And the traveling system, K4 comparison, is being linked to the SIR. And we see the result we obtained since 2009. And we were happy to see one of the more recent results from the PTB, which is a very good agreement with all the results in almost 20 years ago in the SIR. We have also a remote mode since 2021 following the pandemic. So we have accumulated over time 20 on-site comparisons, 16 NMIs, 5 regions. And for the radionuclides as listed here, we see fluorine-18 for PET scan, technetium-99 I talked about before. We plan to use it for manganese and gallium. And in longer term, probably also those 3 nuclides here. Many laboratories worldwide, the BIPM cannot afford all these comparisons, not possible. So in 2023, the CCRI agreed that RMOs would develop and run copies, 30 comparison, linked to the SIR through the BIPM 30. So now, in recent year and in future, we will go on making knowledge transfer about the CIRTI to the regions to support them as far as we can. And about that, I see next talk by Rafael, who will say more about it. And then finally, of course, we are in the move also to make digital transformation. So the SIR reports are digital for a few years now. We are using digital electronics for the ESIR and in development for the CIRTI. However, I'd like to say that there is the issue of storage of large amount of data and also, of course, of the validation of the data analysis software that you need to do, to use. And so the BIPM has been developing a platform comparison of software that's more or less ready, needs to be maybe tried and put in available to the users. So the conclusion is that the BIPM developed new instruments when new needs were identified. With a small group of 4 or 5 people and with the support of secondees, the BIPM has been maintaining, developing, and upgrading to new technologies the instruments for the K1, K4, and K5 continuous comparisons since respectively 1976, 2009, and 2024. So I thank you and the NMIs and the CCRI for your support.
Thank you for that introduction, and thank you to Karin, who's going to make my talk much shorter because she's introduced a lot of these things I'm going to show you, at least in terms of the introduction. I'd also like to say ahead of time that I'm representing content from many people in the room and from around the world. So there's the boring title, but then I thought, let me come up with something else, and I call it the fast and the radioactive. So I thought, well, before I can tell you the story of the CIRTI, I have to tell you about the SIR, and you heard a great introduction from Karin, so I won't belabor this point. It stands for the Système International de Référence, and it's basically provided the basis for measurement traceability and allows us to ad hoc compare with each other around the world. And so as Karin described, you can send an ampoule, a sealed, flame-sealed ampoule to the BIPM, and then it can be measured there in the SIR and given equivalent activity and compared with other samples that are sent. And we were so successful that it was then extended to the ESIR. But I want to first talk about another short story that maybe a lot of people at other NMIs can or DIs can relate to. And that's of something that we had to do. Once upon a time, there were 3 ampoules of yttrium-90, and we had to send them in consecutive weeks from Ottawa to Missouri in the United States, 1 each week for 3 weeks. 2 ampoules ended up getting stuck in Memphis, of all places, And the third arrived within 24 hours and leapfrogged both of the other two. All the shipments had the same courier and the same paperwork. So there's virtually a roll of the dice. It's Monte Carlo as to whether your sample is going to arrive. The same story is for others that at least I have tried to send from Canada to the BIPM over the last 15 years. Fortunately, some of them have very long half-lives, so that's okay, and they could get stuck. But it's either slow or costs a lot of money, so not so fast, and it made me furious. So hence the traveling instrument, or I'm sorry, the transfer insert, but I've called it the traveling instrument before because that's precisely what it does. It goes around the world. And it's a transportable system that allows at least those RMOs that are a bit more geographically separated from the BIPM to be able to demonstrate their capability for measuring these very short-lived isotopes, most of which are in demand for the medical community. So how does it work? Well, the CRT starts at its home, and it's kind of related to its parent, the SIR, and there's a link that's measured here, and then it travels every so often to the surrounding RMOs and then returns home. So the CRT stats, I think these are from 2023, so I think there were a more updated on Karin's information, but essentially it's had a huge take up, so much so that it is in extreme demand. And so what the difference is that we thought, well, what in essence is happening here is we need to increase this frequency for the far-flung RMOs, that being SIM, those in the Asia-Pacific, APMP, QMET, and even AFRIMETs. And as we see, even in Euromet, where you have such a concentration of NMIs, it's hard to get the CRT to come to your lab every so often or frequently. So it's allowing us to be a little more inclusive. It improves the efficiency and decentralize things. So the idea is, can we each have our own RMO? And so how would these work? So again, the BIPM CRT is linked to the SIR. And in a similar way, we now can share the workload. So if you have already a couple of labs that are in your RMO that have participated in BIPM CRT comparison, those are then participate in a round robin within that individual RMO. And then the question is, how do we link? And it's pretty obvious at that point. Essentially, there's the first link from the SIR to the BIPM CRT, and then there has to be yet another one to those individual RMO CRTs. So in order to increase robustness, the decision has been made to, say, have two links and take the average of those, again, to make sure that there isn't any individual bias that you would have here. Again, the hosts are not necessarily a CRT pilot lab itself. So it can be wherever the CRT in your RMO is traveling at the time. So what is the price to pay? The price to pay, at least for metrologists, is there's now two links. And as we know, as you have traceability chains that get longer, the uncertainty never improves. It always increases. However, in this case, we're talking about a very small increase. You don't have to really look at the numbers, but essentially it's small. It's very small compared to the typical uncertainty of the primary standard. And hence, essentially, most of the time, it doesn't even affect the final analysis. So I always look at it in another way. I mean, Karin seemed to indicate that, yes, they're not too busy. That's not the case. They have that as well as a plethora of other things to do. So I always think about the BIPM is very important to all of us. So we need to make sure that they get the resources they need, and always try to see, can we reduce the burden on them? And I think the RMO CRTs are going to hopefully have this effect. And so the BIPM CRT is not going to be retired. It essentially will now can go to the RMOs which don't have a CERTI at all. So for now, that would be AfriMets or maybe CUMET. And they also will go to pilot labs alternating in the different RMO, as is the current practice. So in order to make sure that they're getting that link performed. And as usual, they're very benevolent and caring and like a very good parent. And they are always saying that this is going to be still, the BIPM CERTI is still going to be available to go to those RMOs, which are those pilot labs, to see if we can figure out if there's a problem that can be solved together. So I think you get the picture. The RMO CERTIs are going to compare within their own region. And then one condition, though, is if you're developing it, and of course, you don't want to keep it for yourself, you need to help your region. We are all stronger, the more of us that are involved in metrology. My personal opinion, and this is the caveat, is nobody else like myself, but another alternative is, occasionally every 5 years or so, all of the RMO CERTIs can come here and we can all do a massive campaign. But of course, that means that the onus is on us as RMOs to make sure that we send a qualified individual, not to dump it all on the BIPM to do that for us. So as this idea was developing, we thought, let's come up with a different criteria for these RMO CERTIs. This was done in a very thoughtful way. And I'm not going to read the different criteria, but I'm going to highlight a few that were recommended. You want to have as close a copy to the BIPM CERTI as possible. That makes a lot of the linking and a lot of the other uncertainty determinations a bit easier to determine because everything should be in principle the same. You want one copy per, the BIPM provided one copy of the shielding, the liner, and the stand. So each of the RMOs is provided that by the dutiful parent, the BIPM. I'll emphasize a threshold energy here, which is for the CERTIs set by niobium 93 X-ray peak. However, making this niobium source is extremely difficult. So it's got a very low cross-section and a very long irradiation time to get anything significant. So there might be and are other potential alternatives to using this isotope. The other thing is a reference source. Just as the SIR has used these historical radium-226 sources, we need also one to ensure that we've got that long-term stability. And again, here there is a alternative to use, and that's holmium 166 metastable. And so those are the items there I wanted to point out. And now, thanks to the previous speakers, both Horan and Karin, and who have mentioned Monte Carlo, there is potentially here something else that we can do, and that'll be in my last slide I'll talk about. We all love the key comparison database, and so perhaps now we need an SCDB. So let's go to the different RMOs and see what the status is. In my RMO, the pilot lab taking care of this development is the IRD, LMRI IRD in Brazil. And what you see here is basically the entire RMO CERTI, including a copy of the same CERTI detector. And in this small picture here is the longevity sources that they've decided to use, which is the 166 metastable that I mentioned before. And so where are they at? Well, they've assembled the detector, as you've seen. They've gotten several of the amplifiers and all the readouts going. They're doing current testing with cesium-137 and cobalt-60, and they'll investigate different softwares and MCAs in the coming months. And they're hoping to, at least in this stage, have it ready by the end of January 2026. And I think they've done an incredible job. But there is another maybe sequel. It's not always better than the first, but and this is probably the case here. NRC also purchased the detector. Not that we're going to make yet another CERTI, but we thought there's something that we can assist our colleagues at IRD. Perhaps we can do Monte Carlo, which we are doing. Perhaps we can develop another readout system that we can share or compare with our colleagues at IRD. At the very least, we gain experience, which will facilitate the comparisons that we anticipate to have with the SimCERTI. And so one of the things that we have in our region, or at least expertise that we have, is by using the EXNRC Monte Carlo code. And you can see an example of a distribution data versus the Monte Carlo simulations, very good agreement. And we are seeing a little bit of an offset there that we have to sort of evaluate. And I've gotten some good ideas of where that could be from the BIPM CERTI. The other thing, as I mentioned, that we're doing is we're also using different digitizers and different digital pulse processing conditions with different CAN pulsars to use as potential alternatives or comparative ones that we can use. And this is a user interface, graphical interface that's written by my colleague at NRC. What about the APMP? Well, the APMP's gone full hog, and they're developing multiple CERTIs. So all the NMIJ in Japan, NIM in China, and ANSTO in Australia, they're all developing a CERTI of their own. ANSTO has acquired similar hardware to the BIPM. And interesting enough, they've also looked at producing the stability source and the threshold source of niobium because they have a nice on-site reactor. So that's really good. However, it still remains to be seen. There's still some development here because, of course, there might be some impurities, and whether we can deal with that or they can compensate with that, that'd be very interesting. NIM is already testing a link between the APM CERTI and the BIPM CERTI. And finally, NMIJ has conducted a bilateral comparison with another NMIDI in their region for fluorine-18. So very good progress in that region. Now, multiple CERTIs, what are the advantages and disadvantages? I think certainly having redundancy is never a bad thing. Sharing the workload is something we keep saying, sharing the workload, reducing the workload. Sometimes that can maybe be on both ends of the seesaw. You're sharing the workload, but now you've got three, so you've got to do 3 times the work just to make sure that all things are linked. Again, no judgment here, it's just perhaps advantages and disadvantages. I think developing devices in parallel, some close to the original CERTI, and others potentially incorporating other ideas. It's beautiful because it allows us to achieve both reliability and innovation at the same time. And again, maybe the disadvantages can be looked upon as advantages depending on your point of view. But of course, you have to now link with multiple CERTIs with the BIPM CERTI. But again, that can all be done in one place, maybe a measurement campaign, so all done at once with the same source. And again, just to make sure that there is equivalence is maintained. And then finally, the sort of spin-off. There's a Euromet proposal, and it's coordinated by NPL. And they're planning not to have a copy, but to use very similar geometry, but use a cerium bromide detector instead. Has better energy resolution. It's a faster detector, so perhaps they can accommodate higher rates. And they also mention building comprehensive Monte Carlos. They plan in their proposal to have training sessions. They have very good training sessions as normal with all these projects. And they will set up, their plan is to set up a new working group under the Euromet TCIR, which will retain the stewardship of this device. And so finally, I will just give a little bit of a advertisement to an idea that was described in a very recently a CCRI webinar on challenges with Monte Carlo simulation and ionizing radiation from my colleague Frédéric Tessier. And that is to potentially create, and it's not even potential, it's there. He's created this Git repository, and the proposal is that we need to have vetted Monte Carlos. So you can, in principle, just like you have in the KCDB, you would have an SCDB, which would allow us to start with common geometries for common things, just like the SIR and potentially other common detector systems that we have. A, so that we can have vetted Monte Carlos, and then B, once everybody's comparing the same thing, you can then put the different packages on a similar plot as you have currently with isotopes in the KCDB. Thank you.